All right, cell structure function, chapter 4, AP biology. This is a basic overview of the cell and its component parts and what they do physiologically for both, whether it's a prokaryotic cell or a eukaryotic cell. We know that cells themselves were discovered in 1665 by Robert Hooke. He coined the term cells. Basically, what he did is he realized that when he was examining cork cells under the microscope, it reminded him a lot of what his living quarters looked like. Little boxes that they called cells, and that term stuck today. We could also credit Antony von Leeuwenhoek for the development of the microscope. Early studies of cells were conducted then by Matthias Schleiden and Theodor Schwann in the 1838 and 1839. And basically what Schleiden and Schwann have proposed is this thing called the cell theory, mixed in with the thoughts of the hypotheses of Rudolf Virchow. And the cell theory itself is a fundamental theory of biology, just like evolutionary theory, germ theory, or endosymbiotic theory. The cell theory essentially states that all organisms are composed of cells. Cells are the smallest living things. They are the basic unit of structure and function, and cells come from pre-existing cells. All cells today do represent a continuous line of descent from the first living cells on our planet. So those are the three basic statements of the cell theory. All organisms are composed of cells. Cells are the smallest units of living thing, maintaining both the structure and function, and cells arise from only pre-existing cells. Cells themselves are limited in size. Most cells are relatively small due to the reliance of diffusion of substances in and out of the cell. Remember, diffusion is a type of transport across the cell membrane. When we talk about the cell itself, the cell membrane would represent the surface area of the cell, and then the amount of space that the cell would take up would represent its volume. So when we look at the rate of diffusion, we could say that the rate of diffusion is affected by surface area availability. So the amount of area around the cell or the cell membrane to allow for diffusion to take place. Remember, a job of the cell membrane is to control what goes into and out of the cell, because it is going to be a barrier between the external cell and the internal cell. Rate of diffusion is also affected by temperature. Remember, the true definition of temperature is the average kinetic energy of the particles that make up the substance. So when we think of looking at a rate, we're looking at some sort of speed or motion. So the rate of diffusion will be affected by temperature. You know that kinetic energy increases as you increase temperature, or you add heat to a substance, and vice versa. If you were to remove heat or release heat from a substance, the substance would slow down. You also know the rate of diffusion is affected by the concentration gradient. So the concentration of particles across the membrane. And we know that in diffusion, the greater the concentration of a substance on one side of the membrane and the other, we know that things will readily diffuse across the membrane from areas of high to low concentration. And the gradient itself would also be the space or the volume to which those substances would need to travel. So here you can see basically the cell radius, surface area volume, and surface area volume ratio. So the surface area to volume ratio, organism made of many small cells has an advantage over an organism composed of fewer larger cells. Basically, as cells increase in size, its volume increases much more rapidly than its surface area. So the space within the cell is going to increase more rapidly than the actual surface area, and the surface area would represent the amount of space that you have for things to cross in and out of the cell. Some cells overcome these limitations by being long and skinny, like neurons, because some of the larger cells in the human body, some of the longest cells are neurons of the nervous system. Not many cells are visible to the naked eye. Most are less than 50 micrometers in diameter. This here is the unit for micron, so micrometer. The resolution, ultimately then, when we look at microscopes, the resolution is that minimum distance, which 2 points can still be apart from each other and still be distinguished as 2 separate points. Objects must be 100 microns apart for the naked eye to resolve them as 2 objects rather than one single object. 2 basic types of microscopes are the light microscopes and the electron microscope. Light microscopes use magnifying lenses with visible light, resolve structures that are 200 nanometers apart, limit to resolution using light. The electron microscope ultimately uses a beam of electrons, and those can resolve structures that are about 0.2 of a nanometer apart. The 2 basic types are the scanning electron microscope and the transmission electron microscope. Transmission electron microscopes are going to transmit electrons through the material to ultimately give you an internal image. And the scanning electron microscope is going to beam electrons onto the surface of the specimen, giving you a surface image. So here you can see in this chart here, I'm going from 1/10 of a nanometer all the way up to 100 meters. You can see where about the human eye, the light microscope, and the electron microscope falls with viewing cells, molecules, all the way up to the macroscopic human organism. So if we look at cells, basic structural similarities with all cells have DNA, whether it be in a nucleoid or nucleus. All cells do have DNA. All cells have a cytoplasm region, the semifluid matrix of organelles or cell structures, more importantly, and cytosol. All cells are going to have ribosomes as well, and ribosomes are where you have the site of protein synthesis, and protein synthesis is the process that is going to make or synthesize proteins. And all cells have a plasma membrane. And the plasma membrane is going to serve, or the cell membrane will serve, as a barrier between the internal and external cell and regulate what moves into and out of the cell. So all cells have a plasma membrane, and the major constituent of any cell membrane are phospholipids with their hydrophilic heads and hydrophobic tails. And because cells are mostly water internally and externally, you have a high aqueous environments. We know that those phospholipids are going to form a bilayer where the hydrophilic heads will face the aqueous water environments and the hydrophobic tails are going to hide or pull themselves away from those aqueous environments. Prokaryotic cells are the most primitive types of cells. The simplest organisms that we would say are the bacteria. They lack membrane-bound nuclei. We know that because they lack membrane-bound nuclei, we say that their DNA is present in the area called the nucleoid. They also have a cell wall outside of the plasma membrane. They do not contain ribosomes. They do contain ribosomes. And the reason they contain ribosomes is because ribosomes are not membrane-bound organelles. And ribosomes are important because they are going to manufacture proteins. Remember themselves that ribosomes are RNA subunits that are going to come together, and RNA itself is a nucleic acid. So that's why all cells, whether it be prokaryote or eukaryote, are going to contain ribosomes. There are 2 domains of prokaryotes. There's the archaea and the bacteria. We are more closely related to the archaeans than we are the bacteria in the lineage of life. The Archaean bacteria, if you recall, are those extremophilic bacteria. The bacteria is going to be the true bacteria, the bacteria that we are most familiar with, like Salmonella, E. coli, Bacillus, Streptococcus, all those. If you look at it, here is the typical structure of a prokaryotic cell. The prokaryotes may or may not have some of these structures, like the pilus. The pili are going to be used for attaching to surfaces. You also have conjugation pili, which are going to play a role in sexual reproduction of bacteria cells. The cytoplasm, the ribosomes are going to be the site of protein synthesis. The nucleoid is the area where the DNA is stored. You have a plasma membrane, the cell wall, and a capsule. A capsule is also known as the glycocalyx. That's going to be surrounding on the outside of the cell wall, and then a flagellum is used for locomotion. Bacteria cell walls themselves, most bacteria cells are encased by a strong wall composed of peptidoglycan. Peptidoglycan is in the cell walls of plants, fungi, and most protists are going to differ in that sense. It's going to protect the cell, help maintain its shape, and prevent excessive uptake or loss of water. When we think of bacterial resistance to antibiotic or susceptibility, it often depends on the structure of their cell walls. So when we think of the true bacteria have peptidoglycan in their cell walls, and the extremophilic bacteria lack peptidoglycan in their cell walls. So the archaeans are going to lack it, and the true bacteria will have that peptidoglycan. Flagellum themselves are present in some prokaryotic cells. You may have one or more or none at all. Flagellum are used for locomotion, so that would be movement of the cell. And basically what happens is you get this rotary motion of the flagellum. So it's a circular rotation, and it's going to be able to propel the cell in direction. When we looked at the debate of whether evolution should be taught in schools because it is a fundamental theory of biology, when we looked at the case studies of evolution versus ID or intelligent design, the mechanism for which flagellum would work was one of the key components in supporting evolutionary theory. There are many, but this was one of them that they used. So if you look at the typical flagellum, you do get this motor rotation, which you could see here. You have the hydrogen ions that are moving across the membrane there. The flagellum, you have an outer ring and an inner protein ring. Those are going to be embedded into the plasma membrane or the cell membrane of the bacteria. Then you have that cell wall, or the peptidoglycan portion, and then you get an outer membrane there. So you get these motors that are going to run, and ultimately it causes this whip-like rotation within the flagellum itself for moving or propelling that bacteria slide. Eukaryotic cells are going to possess membrane-bound nuclei, and they're going to be more complex than the prokaryotes. Basically, we see that they have membrane-bound organelles, and this basically leads to compartmentalization. That is the hallmark of eukaryotic cells because they have these cell structures or organelles that have membranes and are going to perform a specific function. They also have a group of structures that are going to play a role in this whole thing called the endomembrane system. The endomembrane system we will look at a little later. We'll look at a little bit further in this chapter. We'll discuss the key structures that play a role in the endomembrane system and what they do. Eukaryotic cells also possess a cytoskeleton for support and to maintain a cellular structure, much like our endoskeleton does. Our skeletal systems provide support, and it helps give us structure, the structure of the human body, which will be our bones. That's what the cytoskeleton is going to do for the cell as well. It's also going to serve as a highway system. So if we look, here's the typical round shaped animal cell. It may or may not have all these cell structures. Some cell structures, depending on what type of cell it is or differentiated into, will be more prominent in certain areas of the animal than in other areas. And we'll talk about some of those as we get into the cell structures. The other eukaryotic cell, this is typical of a plant cell. The plant cell here you could see, again, these cell structures may or may not be visible, but this is a typical plant cell and a typical animal cell. And basically when you carry it out over to, I'm looking at fungi and protists, who also have eukaryotic cell types, by basically covering the typical plant and typical animal cell, you could pretty much know what's also in a fungi or a protist. So basically what makes a eukaryotic cell eukaryotic is that it has a nucleus. This is where it's going to store the genetic information known as DNA. Most eukaryotic cells possess a single nucleus. Also embedded in the nucleus, you have the nucleolus. The nucleolus is the region where you're going to have the assemblage of ribosomal or rRNA. So you will make ribosomal RNA there. Surrounding the nucleus is an envelope, which, like any cell membrane, is a phospholipid bilayer. So you have two phospholipid bilayers in the nucleus. And because you have materials that need to go into and out of the nuclear region, such like messenger RNA, ribosomes that are made, etc., the nuclear envelope needs to have pores in it. So you have that nuclear pores in the envelope, which is going to control what goes into and out of the nuclear region. In eukaryotes, the DNA is divided into multiple linear chromosomes. Chromatin is a chromosome plus protein. When we looked at DNA packing, or you might have learned about DNA packing in advanced biology or a first-year biology class, you know that DNA is wrapped around special proteins called histone proteins. And basically, what will happen then is chromatin it will condense during cell division to form structures that we are more commonly familiar with, called the chromosomes. So if you look, here is the typical structure of the nuclear region. Here is the nucleus. You have the chromatin. You also have this outer and inner membrane double bilayer here that will make up the nuclear envelope. And then the nuclear pore has these cytoplasmic filaments, and on the inside you have this nuclear basket. Ribosomes, these are going to be the site for protein synthesis. It's found in all cell types in all three domains. Ribosomes are made up of ribosomal RNA called rRNA, which is a protein complex. Protein synthesis also requires messenger RNA or mRNA that is going to be made from copying the DNA, the gene, and then you also have transfer RNA or tRNA that is going to transport an amino acid. And we'll study that in greater detail when we get to protein synthesis. But ribosomes may be found free in the cytoplasm, or they may be associated with internal membranes like the endoplasmic reticulum. The endoplasmic reticulum, when it contains ribosomes, is what you would call the rough endoplasmic reticulum. Additionally, you have this endomembrane system. It's one of the most fundamental distinctions between the eukaryote and the prokaryote cell. The eukaryotic cell and the prokaryotic cell, prokaryotes lack an endomembrane system. Eukaryotes have this endomembrane system. And like it says, endo and membrane is a series of membranes throughout the cytoplasm. So a series of membranes that are found within the cell itself. It's going to divide the cell into compartments where different cell functions are going to take place. A lot of this will be when we look at the synthesis of materials or macromolecules, packaging those macromolecules, modifying them, shipping them out, also making energy and breaking down some products that are produced via metabolic reactions. One of the parts of the endomembrane system is the thing called the endoplasmic reticulum. There is the rough endoplasmic reticulum, or RER. It makes it rough due to the attachment of ribosomes to the membrane. Basically, the rough endoplasmic reticulum is involved with the synthesis of proteins that are to be secreted. So what will happen is they're sent to lysosomes or to the plasma membrane. And those proteins, if we talk about like a amylase or another digestive protein that would be manufactured via the rough endoplasmic reticulum, go to the Golgi apparatus where it's going to be modified and packaged, and then it will be shipped via transport vesicle to the external cell where it will be secreted, which is a type of transport called exocytosis. And we'll look at that entire process in chapter 5. The other type is the endoplasmic reticulum smooth. Smooth ER or SER has little to no bound ribosomes to it. It has a variety of functions. One major function is the synthesis of materials, especially that of lipids. So smooth endoplasmic reticulum does synthesize lipids like steroids, so estrogen, testosterone, progesterone. So they're often, the cells that make up our gonads, testes, and ovaries are going to have a greater abundance of smooth endoplasmic reticulum. Smooth ER also plays a role in drug detoxification or detoxification itself. So our liver cells are going to have a greater abundance of smooth endoplasmic reticulum. It also stores a lot of calcium 2+, and we know that the storage of calcium 2+ is important for bone. It's also important for muscle. We'll look at a little bit of that with the muscular system and contraction. So the ratio of rough ER to smooth ER is going to depend on the cell's function. If you look at self coming off of the nuclear envelope, you will have the rough endoplasmic reticulum and you will have the smooth endoplasmic reticulum. Beyond that, then, you have a structure called the Golgi apparatus. The Golgi apparatus is flattened stacks of interconnected membranes. We sometimes call it the Golgi bodies. Basically, the Golgi apparatus is going to function in packaging and distributing of molecules that are going to be synthesized at one location and then used at another location within the cell or even outside the cell. So when we think of the endoplasmic reticulum synthesizing molecules that are to be secreted, those molecules would either go to the lysosomes, which are going to play a role in digestion, or they'll go to the outside of the cell via exocytosis and be expelled and play a role somewhere in that fashion. The Golgi apparatus has a cis and a trans phase, and we'll talk about that because that's going to play a role with what's going into and out of the Golgi apparatus. Basically, the vesicles transport molecules to their destination. So if we look, here is a transport vesicle, here's the cis phase, here's the trans. If we think about the packaging of materials, here you have the nuclear envelope. the rough ER, the smooth ER. So if we have a membrane protein that has been synthesized by the ribosomes there, it gets packaged in this transport vesicle, and the transport vesicle is going to come in. So the vesicle containing proteins buds from the rough endoplasmic reticulum, and it's going to diffuse through the cell and fuse to the cis face of the Golgi apparatus. So the cis face is going to face the endoplasmic reticulum. The trans face is going to face away from the endoplasmic reticulum towards the cell membrane. So basically what will happen then is that vesicle will fuse to the Golgi apparatus, and then that protein will be expelled to the inside of the Golgi apparatus, where it's going to be modified into what it needs to be doing, and then it's going to be transported out via a vesicle, and that vesicle will be released from the trans face of the Golgi body. And now at this point, we go from using a transport vesicle to a secreting vesicle, and the vesicle may travel to the plasma membrane and release its contents to the extracellular environment. And you can see that going on here. So the last structure we'll do for today is lysosomes. These are membrane-bound digestive vesicles. They arise from the Golgi apparatus. The lysosomes are going to contain enzymes that are going to basically break down macromolecules. So they're going to catalyze or speed up the breaking down of the larger macromolecules. So the polypeptides, proteins, polysaccharides, etc. So we would say that this is a hydrolytic reaction or a catabolic reaction. It's also help destroy cells. It's also going to help destroy foreign matter that cells have engulfed via phagocytosis. So they play a role in digestion. So if you look, basically phagocytosis will bring something into the cell, cellular eating. That type of vesicle will be a food vesicle then, and it will come in and travel to a lysosome, and the lysosome will digest those products. Because inside the lysosomes, you are going to have in there enzymes that have been manufactured that are going to contain, play a role in the hydrolysis of macromolecules. All right, so that's it for today. We will finish the cell structure function chapter 4 in our second lecture. Thank you for watching, and if you have any questions, please have them ready to ask in class. Enjoy your day.